Yeah, so today we are here to sample some sticky traps and some pitfall traps from one of our trap locations in these plot to field sites. These plot to field sites are really great because we have 4 different treatments going on, so we're able to really kind of compare between the treatments. Right now I believe we're standing in a canola monoculture, but we also have pea monocultures and 2 intercrop treatments, one with no fertilizer and one with half the recommended dose of fertilizer.
We have 2 different traps here. So Jose, what is this?
This right here is a yellow sticky card. It's pretty much little cardboard, little sticky on it, and it's really great for passively sampling insect populations. So if you can we'll get a close-up after, but we have here a bunch of flies. So we'll kind of leave these out for 2 weeks, capture as many insects as we can, come back and sample them. We'll bring it to the lab and get a better look at what's actually on these sticky cards.
So what you're hoping for is as the crop grows, then your bug population kind of changes.
Yeah. So bug populations are going to change depending on the crop stage. So as the crop grows, like you mentioned, we will be seeing different bugs appearing, which is good for us because we can kind of see how things are changing in each of these treatments throughout the season.
And you're hoping for an intercropping, you'll have a whole set different.
Yeah, that's definitely sure. We're looking to hopefully have increased diversity and abundance of parasitoids and natural enemies. Because what we're trying to sample with these yellow sticky cards are going to be diamondback moths, flea beetles, and unlikely, but maybe, some pea leaf weevils.
I wish you a lot of bugs.
Yes, here's hoping. So here we have a field pea plant. It has been uprooted, but you can see the nice pretty plant we have going on here. Now, pea leaf weevils, as their name suggests, love field peas. And the characteristic damage that they have is notching. It's very characteristic and very easy to tell apart. So when we're going through all these treatments, we can sample some field peas. We don't have to take them up, but we can just look at them, count how many nodes there are, and count how much notches we see per node. The node of a field pea is the 2 little leaves. Each of that is 1 single node. Pea leaf weevils prefer field peas that are from the 2nd to the 6th node stages. These ones are a little bit...
we're kind of late.
Yeah, a little advanced, but we're still sampling. We're still doing our best out here. Another interesting thing is at the roots, normal field peas will have a lot of nodulation happening at the roots due to its symbiotic relationship with rhizobium bacteria. The larval stages of field peas target the root nodules, eating them up, and remove or eat away at those nodules, preventing the peas from be able to fix nitrogen. So both the larval stages and the adult stages are important to monitor for. But after the field pea reaches above the 6th node stages, the notching is not causing significant damage to the field pea plants. They're able to sort of compensate for that.
And this will also help you to decide, after you calculate how many notches and how much of damage you have seen, if it's wise for the farmer to spray or not.
Yeah.
The economics of it.
Exactly. Because we have all these plots side by side, we're able to directly compare, in what this case is here in intercrop, to, a couple rows down we'll have a pea monoculture. So we'll see just how much changes we're seeing from plot to plot.
Right.
In the back here, we also have pitfall traps. This is pretty much a hole in the ground with some plastic cups, and these can be dry or wet. Here we have a wet pitfall, and this is filled up with a little bit of antifreeze, as you can hopefully see there. And what we're hoping to capture with these ones is terrestrial bugs. So bugs that primarily disperse by just walking on the ground. So we're hoping to catch some pea leaf weevils and some natural enemies. Hopefully you can see, but we have a couple spiders in there already, which is great. Spiders, carabids are all really great natural predators of many insect pests. So getting an idea of how these populations differ between the treatment sites is also very important to understanding the importance and potential for Piola intercrops to increase the diversity and ecosystem services such as predation. What we have here is one of the sticky cards. I just took it out of the tie, and we can see we have a bunch of insects on there. We have a hoverfly and 1 lady beetle and a couple spiders, so that's kind of good. We kind of went over all the traps and what we're doing for one trap location, but we also have a second trap location 25 meters into each of the treatment plots. So we'll essentially be having an interior trap and an edge trap. And we'll also be comparing between the insect communities that we find in the edge and the interior, as that can definitely have an impact on what we're finding.
So we have pea and canola growing quite tall, as you can see. It's mid of July, and I have Jose from University of Alberta with me collecting some data, and he'll let us know what he has been up to ever since we saw him last.
Right, so last time you saw me, we were doing some pea leaf weevil damage assessments. Behind the scenes, we were doing some flea beetle damage assessments when the canola was at the earlier stages, so dicot to 4-leaf stage. Right now, however, we are conducting sweep samplings. So right here with me I have the trusty sweep net, never leave home without it. And what we'll be doing is sweeping the canopies of this intercrops and the monocultures here and all these treatments, and sampling the insect communities in each of these plots. The great thing about the sweep nets is we're actively sampling. So we're traversing the plot and sweeping and hoping to get much more insects than we will get from just the sticky traps, and also different insects than we would be getting from just the sticky traps themselves.
And what I saw from your collection today, we have pollinators, we have good insects here, so all
Yep.
our helpers, and then we found a diamond black moth.
Yes, so the thing that we are looking for is the diamondback moth, and that's actually what we found. So these sweep samples, along with all the other samples, we're going to be looking for parasitoids, predators, pollinators, all the beneficial insects that are going to be helping control insect pest populations, but also help pollinate. And surprisingly, and kind of lucky for us, we managed to find a diamondback moth, one of the major insect pests that we're going to be looking at.